Okay. So tonight we're going to hear about the biology of love from Dr. Stephen Phelps. So let me tell you a little bit about Stephen. He grew up on a farm outside a small town in Illinois called Ava, Illinois. And as Stephen points out to me, that's one of the rare places in Illinois that's a palindrome. So you've already learned something tonight as we go forward. And Stephen was always interested as a child growing up on this farm in just how things worked, including the animals. And he was also the first in his family to go to college. And he thought always that he was going to grow up and study in college and become a biomedical engineer, but he didn't do too well in that major. But he had to take some biology courses for that. And in a biology course, he learned all the magic about evolution. He learned all the magic about how the brain works. And he thought, what if I combine those two fields of study, and made that my career? And so he was off and running. And fast forward to now. What Stephen is professor in integrated biology here at UT. And he's also director and co-founder of UT's Center for Brain Behavior and Evolution. So what a long way he's come from those very beginnings. Please welcome Dr. Stephen Phelps.
Well, thank you very much, Jay, for that kind introduction, and to Jay and Angelina for making this event happen 140 times. Wow, that's amazing, isn't it? And thanks to all the folks who are here tonight and taking part in this Texas Science Festival. It's wonderful to see so much enthusiasm for the science going on here at UT. And those singers, there are so many songs about love. I mean, thank you. I think accidentally in love is probably the best way to fall in love, isn't it? How many folks here have been in love or are currently in love? All right. I'm not going to ask how many have not, but it's a pretty common experience. And you think about all the songs that are written about love. Had to be you. Well, maybe it didn't have to be you, but there are a lot of songs about it. It's pretty important to us. So as Jay said, I'm an evolutionary neurobiologist, and I'm really interested in animal behavior, including human behavior, and thinking about how it evolved and how it evolved at a really kind of nitty-gritty, detailed level, at the level of brains and molecules. And so I'm going to tell you about some of that work. But before I do, I want to just kind of give you 3 takeaways. And the first is I hope to convince you that the capacity for love and its complement in other species, bonding more generally, is an evolutionary adaptation. It's something that helps us collaborate with one another. Secondly, I want you to realize that there are mechanisms in the brain, in our brains and in the other brains of species, that we share in common. That is, aspects of bonding and love rely on similar processes and similar mechanisms across species. And the third thing I want to tell you is that despite these commonalities, there is variation in our experiences, again, across species, but also across individuals. So I'm going to start the story tonight at one of my favorite places, which is the Smithsonian Museum in Washington, D.C. Now, they have the Hall of Evolution there. Has anyone been to the Hall of Evolution in the Smithsonian? Yeah, some of us have. It's a beautiful, wonderful place. This is a Neanderthal skeleton called Shanidar 1, a pretty famous skeleton. And one of the reasons it's famous is you see that the right arm here has been withered away from disuse. Now, this is an individual who had a severe brain trauma to the left side of his brain when he was young, but grew to the age of roughly 40 or so, in Neanderthal years, I guess, I don't know. I'm showing my ignorance about that. Grew to the age of roughly 40 or so and was able to do so despite this impairment at a time, 40,000 years ago. We can see this also in still older fossils. This is a Homo erectus, 1.8 million years old. And you see, notice the teeth. This is the skull of a very old person who's been able to grow to an old age. Again, presumably with the help of community. This is what bonds do for us. This is why they seem so essential to our experience. And we can see this not only in fossil remains, but we can see it in the behavior of species alive today who share much of our DNA. So these are two teenage adolescent chimpanzees, not literally teenage, adolescent chimpanzees named Baron and Pee-wee. And this is a photograph taken by Aaron Sendell in the anthropology department here. And he studies the bonds between young chimpanzees and how they shape their relationships into adulthood. And what you see here is a kind of tenderness. They move around together, they pick up and go off and do tasks together. It looks in almost every way like something we would call friendship. But the thing is that there are many species that don't have things that look like friendships, that don't have things that look like romantic love. So the orang, for example, is a great ape, so closely related to us, and yet as adults, they're solitary. They don't seem to need the companionship of other orangs. And so we have these differences in experience, even among relatively closely related species. Well, this is a question that, as Jay mentioned, I became interested in when I cut off my mullet and moved to the University of Illinois, where I was an undergraduate in 1988. And having done very poorly in biomedical engineering, a fact I was going to admit tonight, I took biology as part of my training there before I changed my major to physiology in this enormous lecture hall. It had 800 students, which is bigger than my hometown. But the class exposed me for the first time to evolution and to neuroscience and to animal behavior. And I remember sitting in my dorm and thinking, oh, like maybe you could study the brains of animals and understand something about their emotions or about their experience. And it seemed kind of a crazy idea then, but I realize now it's very much the kind of work that I do. Well, this work really has its origins almost a century ago in the insights of a famous animal behaviorist by the name of Konrad Lorenz. He wrote a monograph published in 1935 whose title is translated in English as Companions as Factors in the Bird's World. And Lorenz wrote extensively about, he kind of redefined how we think about instinct. People before Lorenz thought of instinct as this kind of almost like a reflex that was completely thoughtless or completely without experience or feeling. And he had a different idea. He thought that there were aspects that might be like reflexes, but there were other things that were more about the emotional experience of the animals that were tuned in some meaningful way to their environment. And he talked about each relationship that a bird might have as being like a kind of buddy specific to a particular task, the way you might have a knitting buddy or a bowling buddy. He thought that birds might have flock buddies or mate buddies, or companions, as he called them. So this caused quite a stir, but I think one of the things that was the most insightful about it was that he gave us the idea that we could think of behaviors as emerging from aspects of material, emerging from aspects of mind and brain, and that, like an organ, they could evolve in response to natural selection to become a better fit to their worlds. And he thought also that as a result of those differences in their natural worlds, animals must have an exquisite variety of experiences. And he talked about chickens who have different alarm calls for aerial predators versus terrestrial predators. And he thought, well, I bet they have like a really unique feeling of fear for each of those kinds of contexts. And anyway, I find that really moving and really profound. And in laying the foundations for the modern study of animal behavior, he also gave us some tools for beginning to think about why questions. Why do some animals bond and others don't, for example? This is an Australian pipefish, and pipefish, like seahorses, have an unusual reproductive system in which the males brood the young, and in this case, in these pipefish, they brood the young in pouches in a way that's very similar to pregnancy. And in these pipefish, the males and females form pair bonds, and they patrol a territory together and raise young together. And so their bond is really in the service of a kind of collaboration. We can see other beautiful examples in the natural world. This is a poison frog. Now, poison frogs live in Central and South America, and some species are so poisonous that the skin contains enough toxin to stop 100 human hearts. And that's pretty fascinating, but what I think is maybe even more fascinating is the crazy, fascinating behaviors that they do. So because they're toxic, they don't have to have lots of kids that might get eaten. They have just a few. They lay a few eggs in the forest litter, and often in many species, the male tends to the eggs until they hatch. And here we see a father of a Peruvian species of poison frog, and on his back, what is that there? Kids, what is that there? It's a tadpole. Why has he got a tadpole on his back? Because it's a baby. What he's going to do, he's on the forest floor, and then if he were to look up, you would see, maybe the height of this room, the tops of the trees, where lots of bromeliads, these plants that catch water when it rains. And what he's going to do, he's going to climb up that tree, and he's going to find an empty bromeliad to put this tadpole in. And that tadpole is going to be able to eat insect larvae and other things there. And he's going to go back and check on it from time to time. And if the tadpole's hungry, it'll beg. It'll beg by wagging its tail. And if it wags its tail and begs, he will call his mate. And his mate will come, and she will then lay unfertilized eggs in the bromeliad for the tadpole to eat. It's kind of crazy, isn't it? It's really touching, I think. But here's a strange thing about these frogs. Another strange thing about these frogs. They don't actually recognize their young. You can swap one tadpole with another, and they don't know the difference. It must be a very different experience, the relationship that they have to their young than other species. I had a chance to see some of these frogs in the wild when I came to graduate school. I came here to the University of Texas in 1994 and did a PhD. And I worked with Mike Ryan, who's here in the room tonight, one of the greatest animal behaviorists of the 20th century, I like to say, although it's the 21st. We'll have to see, Mike, about the 21st. And I love the work I did. I worked with frogs with Mike in Panama. But I was really interested in brain stuff, and we just didn't know that much about how frog brains worked. And so I thought after I got my PhD that it would be a lot easier to do the kind of work I wanted to do on interesting, weird rodents. So that's what I did. At the time, researchers were beginning to make a lot of progress in understanding the mechanisms of bonding by studying this strange species, the prairie vole. Now, prairie voles are shaggy little rodents about the size of a tennis ball. So they're bigger than a mouse, but they're smaller than a rat. And almost everybody calls them moles because it rhymes, but they're not. Show them some respect. They are shaggy, and they vary a lot in their social behavior from species to species. The prairie voles live in the Midwest, from the northern edge of Oklahoma, well up into Canada, east from about Ohio, west to Nevada, roughly. So they have a really wide range. And one of the things that makes them special is that males and females form bonds with one another. So they will mate repeatedly over the course of a day. And if no one else interferes with that, then after a day, they will form a bond that will likely last their lifetime. So that bond means that the two of them will share a nest together, they will rear young together, and they will have a small, very actively defended territory that they patrol and keep other voles from entering. So they're like the frogs and like the pipefish, collaborating in a way. Now, the advances people were making in studying the brain and its relationship to bonds came about because, well, researchers initially just noticed that these voles were showing up, males and females together, showing up in traps in the wild. They thought, oh, well, maybe they're monogamous, maybe they pair bond. So they did a variety of studies in the lab that actually showed they did. And then they started to look at hormones which are now famous, like oxytocin, and its less famous cousin that we study, vasopressin. And they began to realize that these hormones were really important for these bonds. And then around that time in the 90s, when I was a graduate student here, researchers at Emory started to look at the brains of prairie voles. And the way you looked at hormone receptors in the brains of prairie voles is through what's now a pretty old-fashioned technique. You have a brain that's frozen, and you slice it on essentially what amounts to a deli slicer. So really thin slices, and then you lay those out on glass slides, and then you squirt some radioactive hormone onto them. And you wash off all the excess, and then you dry them. And now hormones act in the brain by binding to little proteins called receptors. And the job of receptors is to sense hormones and communicate their presence to the cells that they're on, to the nerve cells that they're on. And so wherever those receptors are, the radioactive hormone sticks. And the more receptors are there, the more it sticks. And so once you dry it, you can expose them to X-ray film. And the darker the film, the more receptor is there. And so this is a picture I took a few hours ago. I dug out some old X-ray films. And you can see these are two brains sliced from the beginning through nearly to the end, stacked each on 4 microscope slides. And then you can see then what they might look like on an X-ray film. Well, we pick the best ones, and then sometimes we give them color just to make them pretty. But this is what the researchers found back in the 90s. They found that the prairie vole, if we looked at one of these brain sections, kind of midway back through the brain, that it had a lot of this hormone receptor in a particular brain region, a particular part of the brain. And if you compared it to another species, one that does not form bonds at all, they largely lack the hormone receptor there. The hormone receptor is fine, they have the gene to make it, but it just doesn't show up in this particular brain region. Well, this brain region is really interesting. It's called the ventral pallidum, and you don't have to remember that. But what's interesting about it is that it's in a network of brain regions whose names you also don't need to know. But what's interesting about them is that this is a network of brain regions that involves a lower brain region from the brainstem called the VTA that releases dopamine. Now, has anyone heard of dopamine? Yeah. What's it famous for? Reward. It makes you feel good, right? And so, it's, in reality, it's more complicated, but, it's pretty close. So it releases dopamine, and it releases them into these 2 different connected brain regions. One, this up top called the nucleus accumbens, gets sensory information. And so that might be, for example, the sight and smell of a partner, shown here in the lower right. And then the reward helps connect those cues, those unique things about your social partner, to an action, like approach. It's pretty cute, isn't it? That took a long time to make. So this insight that, we now know that it's not just that these receptors show up in these different brain regions. We know that they're causally related to bond formation, because if you activate the receptors in the absence of mating, a prairie vole will form a bond, even without mating, which it normally needs to mate. Similarly, if you block the receptors with a drug, they can mate normally, but that mate doesn't become a bond. So something about having these hormones there maybe takes the normal reward of, cover your ears, kids, mating, and makes it something that becomes enduring, that becomes something that is meaningful and lasting. And this work directly inspired work on oxytocin, vasopressin, and love in humans. And one of the first studies in humans was this really fun study by some famous psychologist, anthropologist, and neuroscientist named Lucy Brown. And what they did was they asked undergraduates, how much do you love your partner? Right? Just on a scale of 1 to 10, how much do you love your partner? I'm not sure what those numbers mean exactly, but I know that they can produce interesting results. And what they found was that the higher the rating, the higher the report of love, the more activity these undergraduates had in these reward regions when they viewed a picture of their loved one. It's kind of crazy. More recently in prairie voles, a friend of mine, Zoe Donaldson, this is kind of some of the really new exciting things that are happening in neuroscience. She took a protein that had been engineered to light up when a nerve cell fires. And when I say a nerve cell fires, what I mean is that a nerve cell has a rapid change in voltage, a rapid bit of electrical activity that it uses to convey information to other nerve cells. Not only that, but the pattern of that activity across a bunch of connected nerve cells can form the basis of a memory, for example. And so she took this engineered protein that lights up whenever a neuron fires, and then she put that into one of these reward regions that we just talked about. And so in the middle here, we see outlines of the different neurons that were being recorded that fired at least once in the video. And then on the left, we see two voles, one of whom has this tiny little microscope mounted on his head. And what she found is kind of extraordinary. She found that these neurons would light up just before the male would approach his mate. It was like she was reading the thought, I'm going to go be with my partner. So this is all fascinating and fun. And when I entered the world of prairie vole research, I had just finished my PhD. I'd done a short postdoc at the Smithsonian. I'm still working with Mike Ryan. And I came back excited to work on prairie voles and to learn some more neuroscience in the process. And I came up with kind of a strange project. I decided that I was going to go back to Illinois, where I was from, the University of Illinois, where I'd been as an undergrad, and catch a bunch of prairie voles there because they're native there. In fact, that's where the original work was done with the prairie voles. And so I went to these kind of open fields like this one, and in the fall, the prairie voles are really abundant. You can also see these beautiful giant flocks of starlings and red-winged blackbirds and other things. And so we would put these traps down, these little, if anyone may have seen them at one of the booths outside, these little traps down, especially around clover, little spaces between farm fields, and I could catch prairie voles. And I just had a simple question. If we go and look at wild animals and look at their brains, how much do they vary? Because surprisingly, neuroscience, usually animal neuroscience, which focuses on lab mice, tries to kind of get rid of all the variation a lot of times. And I had this background as an evolutionary biologist. I was really curious just what kind of variation we might find. And some brain regions were incredibly variable and some not variable at all. And that difference turned out to be interesting. One brain region that I'm going to mention now, talk about quite a bit, was a brain region called the retrosplenial cortex, another one that you don't need to remember. But you can see here, these are two X-ray films of two different brain sections from different individuals at about the same level of the brain. And you see that the animal on the left has a lot of the hormone receptor in this brain region, as well as other brain regions that are dark below. The animal on the right has the hormone receptor in the other brain regions, but for some reason, it lacks it in just this one. And the differences between individuals within a species were as big or bigger than many of the differences we saw between species. And so we were fascinated by this, and that became the seed of one of the first major projects that I would do when I got my own lab at the University of Florida. So I went there in 2002, and it was not long after I got there that I met somebody. I was at a gay bar with my boyfriend, and I met a friend. I ran into a friend there, and he was there with his boyfriend, who I had never met, and I thought that guy was so cute. But he was so shy. Like, he stood up against the wall, and our boyfriends were out dancing, and I was trying to talk to him, and I couldn't get a word out. And I would ask him questions, and he would say yes or no, and that was it. But I remember thinking, boy, if I'm ever single, I'm going to look that guy up. And I didn't see him again for, I don't know, probably 3 or 4 years. But I remember, that first meeting now was 20 years ago, and I can remember the space where we were, where we each stood. I didn't love him then. That came years later, and yet I can remember with incredible detail exactly where we were at that moment. For those of you who are in love, does anyone have a similar experience that you can remember exactly where you met your partner? Raise your hand if that's true for you. Yeah, it's really common. It's really common. And again, it speaks to the importance that love has in our lives. Well, anyway, my husband and I are so different. Like, I'm an extrovert. He's very much an introvert. I love the outdoors. He thinks it needs to be air conditioned. And on paper, we shouldn't work, and yet somehow we complement each other, and our differences bring us a lot of joy in many ways. So I'm now a faculty member at the University of Florida, and my students and I begin to study this question about individual differences. And one of the things we do is we take prairie voles and we put little radio collars that emit beeps at very high frequencies. And so you can see kind of this vole has a little collar here. If I can get my cursor in the right place there. That's a little radio collar right there. And it's just on a little zip tie. And then that's a little piece of corn to give you a sense of scale. That's the aluminum walls of the trap. And so we put a bunch of these animals out in the field, and then we radio track them. We just figured out where they were over time. So a few times a day we'd go out, my students would go out, and figure out where in this plot of land, which is shown here in green, it's about 60 feet by 90 feet, and find out, where they were. And over the course of a few weeks, we could have enough data that we could start to get estimates of the probability an animal would be in a particular place. And from those estimates, we could also figure out where they were nesting and who was nesting with whom. So here in this graph, we're seeing that space in green, and then we're showing just 5 pair-bonded males here on the z-axis. That is, and that axis is the probability of detecting an animal in that point in space. And the wired surface are pair-bonded animals, and the solid surface in red is the focal animal. So I said 5. There are only actually 4 pair-bonded males in this one. There are 5 in another one, I believe. And what you can see here is that the focal male, the male that we're interested in at the moment, is the one with a solid red surface. And you can see he stays pretty close to home. That peak there corresponds to his nest, and he doesn't really intrude on anybody else's territory, and nobody seems to really be intruding on his territory. Well, if we looked at his brain later, and we did, we found that he was one of the guys who had a lot of brain hormone receptor in this memory circuit. This is another enclosure, and here we have somebody else who is intruding on the territories of neighboring males, shown here in the red arrows. And when we looked at his brain, we found that he lacked expression in this memory circuit entirely. So what we think is going on is kind of kooky. What we think is going on is that there are two brain types, one that has good social and spatial memory and one that doesn't. And the good one, those males end up being faithful to their partners because they stick close to them and they don't intrude on other territories because they remember where they've been chased away before. The males with poor memory, we think, are less likely to avoid the places they've been chased off and less able to keep track of their mates. And so we find that they encroach on these other territories more often. Not only that, but they're much more likely to sire young with someone who's not their partner. Well, after we found this, we thought we had enough data to suggest that maybe there might be some genetic variation that was contributing to the differences in the brain. And we did a bunch of breeding studies and looked at DNA and were able to chase down this brain variation to just a couple different letter differences in the DNA. Areas that we found were actually important for the way the brain would work. So we can think of this as a high expressing variant in green and a low expressing one in white. Now that we know what the markers are that predicted the brain variation, we could go back out into our samples and say, well, were these two DNA differences, were they equally successful, or was one more successful than the other? If we looked at just the matings that happened with partners, we can see the relative number of young they sired, which we call their relative fitness. We can see that the high expressing allele does much better in that context than the low expressing allele. However, the high expressing allele never sired young, or never showed up in young that were from some philandering. But what was particularly interesting is when we threw all the data together, we found that actually the two versions were almost perfectly matched. So we looked more closely at the DNA, and when we looked more closely at the DNA, we found mathematical signatures of something called balancing selection. And it's a little technical and a little boring for a general audience, but the point is just that there was variation that had accumulated at these regions of the DNA that was a classic signature of natural selection having kept around these two alternative versions for a very, very long time, much longer than our study. So we think that there are different ways of keeping track of your social relationships in these voles, and that selection likes both of them, that they both stick around because they both work in their own different ways. But we wanted to look a little more closely at the idea that this is really memory, because we knew that the brain region was important for memory, but we didn't actually look at it closely in the context of the prairie voles. And we did a couple studies. This first one is a little technical, but I'm gonna walk you through it. These blue blobs are the nuclei, the center parts of cells that contain the DNA. When a nerve cell has electrical activity, it follows up that electrical activity with a bunch of gene expression, with turning on things that are going to compensate for all the energy the cell just spent, but also are going to help the cell learn new things in response to that activity. And you can see those things that are happening as little bursts of specific gene products that are going to coordinate a whole bunch of things within the cell to help do those tasks. But the different gene products, just by chance or because they're doing different things in the cell, they have different time courses. Some are really fast, like these pink ones here. It's a gene called ARC. And some are really slow, like this yellow one. And if you wait, if the nerve cell has a burst of activity, and you wait 25 minutes and look again, you'll see that much of that fast activity has now moved out of the nucleus into the rest of the cell where it can start to do its work there. The slow one, though, is still getting going, and it's still in the nucleus. So why did I tell you all of this boring cell biology? Not boring to me, but the reason is these facts about the different time course of things that happen in response to neural activity are a way that we can try to get a sense of whether a group of neurons contains a memory. And what I mean by that is we think it contains a memory if you activate it repeatedly and the same neurons are active. And so what we did is we took a male vole and exposed him to a mate for 5 minutes. And then 25 minutes later, we exposed him to his mate again. And so if the neurons that were active the first time he saw his mate were active again the second time, that would suggest that maybe those neurons are actually encoding a memory for that mate. And that would show up as both the fast and the slow as little dots in the same place. The fast ones from the last exposure, the slow ones from the first. And then as another group, we can expose them to a mate, but then follow that with an exposure to a stranger. Now, the data I have on this are not as beautiful as I would like because we had an error in the experiment. But the error turns out to be interesting. The student doing this hadn't worked with the prairie vole behavior before. He'd only done the molecular biology. And about half of the voles didn't actually mate and form bonds. And so when we looked at those unbonded animals, we saw that the percent of neurons that were reactivated was very low. But in both cases. But if we looked at just those that form bonds, we found that the percent of neurons that were reactivated were very low if they saw a mate and then a stranger, MS. But if they saw a mate and then saw a mate again, they were much more likely to be reactivated. So it suggests that this circuit that we've been focused on really does contain a memory for a partner. There's another way we could probe the memory, and that's with a really classic behavior test that was developed by Sue Carter, who first really pioneered the work with the prairie voles in the lab, who discovered the role of oxytocin in the prairie voles, for example. This is called a partner preference test. And the idea is pretty simple. If you have a focal animal, and we're talking about males, but this is all true of females as well, you can put them in a plastic arena, and we use ones that are about that big, divided into thirds, and then you can put each of the females on a collar in the two end chambers and allow the male to move back and forth among the three chambers, the empty one, the one with either of the two females. And again, you can do the same thing with females, and they show the same preferences. But what you see, as our data shows in this particular experiment, is that they would much rather spend time with their mate than with a stranger. If, however, we briefly, temporarily inactivated this memory circuit, then we eliminate this preference. It disappears. So it's as though, like in that moment when we've inactivated this brain region, they can no longer recall who their mate was. Well, we've done a lot of work with reward and with memory. And the more recent work we've done is trying to ask more completely, what are all the brain regions that are active as a prairie vole forms a mate? Because there's a lot of other stuff going on that we're interested in. And so we've done that, and I'm just going to show you a bunch of sections of the brain from the beginning to the end. And what these sections are doesn't really matter. You're just supposed to be impressed. So what you see here is a little bit like an fMRI image of a human brain, where each little dot, where it's colored, corresponds to some significant thing going on there. And the significant thing that's going on is a lot of cells are expressing those gene products that indicate they were active during bonding and not during control conditions. And we see a lot of the same brain regions we already knew were important, but we see a lot more too. We see 68 different brain regions. And I won't go into those in any detail, but I will say that in the 20 years or so since that first study I mentioned of human brains in love, there have been a lot more studies of human brains as well. And so there are a lot more brain regions besides just those involved in reward that we know are relevant to human love. But what I think is maybe one of the most interesting are the ones that are not shared with prairie voles. And I've just highlighted the square from a paper in orange. These are brain regions that social neuroscientists, people who study the brain in social contexts, that social neuroscientists have found are repeatedly engaged in tasks like perspective taking, which is imagining what somebody else is feeling or seeing, in things like empathy, in all kinds of complex models we have of other people, our best estimates of who they are and what they feel at a given moment. And I think this is what distinguishes the experience of a prairie vole in a bond from the experience of human love. So if we go back to some of these animals we've talked about, we can think of almost a gradation of experience. We have something like the poison frog that forms what looks like bonds but don't even recognize their own young. And I call that a plesiophilic bond, like a near bond. Then we have the prairie voles that do a lot of things, in addition to the stuff I've told you, that really sure does look like a bond. They give each other social support. They do all kinds of stuff. So I call those true bonds or euphilic bonds. But then we have human love, which has a lot of the same machinery, but has added on top of that our capacity for understanding, our capacity for sophisticated knowledge of another person. And so I call those knowing loves or nosophilic bonds. And this all brings us back in a way to Konrad Lorenz, to the exquisite variety of experience that he thought animals must have. And I'll take a moment here to mention that Lorenz not only inspired much of animal behavior, but his work on imprinting in geese, where the young form these young geese, when they come out of an incubator or come out from under their mother, the first thing they see, they're going to form an imprint, and they're going to take that individual to be their parent. And Lorenz discovered that that was the case quite on accident, and had a close friend of a gray-legged goose who followed him everywhere for much of her life. And that work then went on to inspire work by Harry Harlow in monkeys, and work by John Bowlby in children. And then Bowlby's work gave us attachment theory, which many of you may have heard of in the context of relationship styles. And so there's been this beautiful history of dialogue between work in animals and work in humans, and it also speaks to the continuity of that experience. Bonds are so important to us that we see references to them, to the varieties of their natures in texts from the ancient Greeks. And in the kinds of loves that they lay out, we can largely see nice counterparts in the animal world. But we can go farther back and see in burials in China or Siberia or Italy from 1600 years to 6,000 years or older, this history of our relationships, our loves, and how much they mean to us. Okay, so I want to close out with some take-home messages, the same ones I began with. Love, I think, is an evolutionary adaptation, and bonds allow partners to cooperate. Love and bonding share common mechanisms across species. And so understanding bonds can reveal how love protects us against stress-related disease, for example, or other useful things we can learn from it. And there are natural differences between species, but also between individuals. And so there are many ways to experience love. There are practical reasons to study bonding. But in the end, I think getting to know another species is a little bit like getting to know another person. There are things we share and things that set us apart. But understanding can be its own reward. Thanks.
I'm sure Steve will be happy to answer some questions.
how are you like confident enough to like go after the stuff you want to do, like the science stuff?
Yeah. Well, how am I confident enough? I don't know. I think, I wasn't always confident enough, to be honest. When I was an undergrad in Illinois, Jay mentioned I was the first in my family to go to college, and I had a lot of failures. I worked too many hours as an undergrad, taking too many classes that were too hard, and it all added up to some bad grades my first couple of years. And so when I finished my undergrad degree, I stuck around at Illinois to do a master's degree, partly to figure out if I was smart enough to be a biologist. And I think that's a very natural thing to feel, and all you can do is sort of go forward following your interests and see where that leads.
Okay, thank you.
Welcome. Thank you for your question.
Why do tadpoles go on their parents' frogs' backs even though they can't be out of water, and how do they breathe?
Well, that's a great question, but they're not out of water for very long. So they're still wet, and they're out of water just long enough for Dad to take them up to the bromeliad. So they're fine that way. But there is no other way to get there.
Good question. Thank you.
Thank you. So quickly, there are different type of bonds we have, like a bond to a parent, a bond to a partner, a bond to a kid, maybe a bond to a sports team, or a bond to a friend, a group of friends.
Yeah.
And you mentioned multiple different brain regions that are activated when our bonds are triggered or so are they similar?
Yes, that is something we do not understand. But when we did this whole brain study that tried to make a definitive map of all the paths by which that were involved in the formation of bonds, and we looked at that list, it was shocking how similar it was to large lists around parental care, around other kinds of relationships, and so we don't, and there are differences. I mean, there's some things that are involved in one kind of bond, but not, or one kind of relationship, but not another. But yeah, I think it's a fascinating question to wonder about. What I imagine to be the case is that there may be some. This is pure speculation, but it won't stop me. What I imagine to be the case is that there are general mechanisms around bonding and cooperation that can get used in any relationship, and those are naturally engaged by interaction. And then there are other mechanisms that are specific to a particular kind of interaction, whether it's sexual mechanisms, whether it's parental mechanisms and feeding. And those specificities give each its own unique flavor, but I think they share a lot of the common architecture. Yeah.
Thank you.
You're welcome.
This is a very similar question to the last one, but how would you define romantic love specifically, and how would that definition differ compared to other forms of love?
Yeah, I mean, romantic love, I think, is love that is in some way sexual in nature. I think that is the gist of it, and that differs from other kinds of love.
Have you measured the difference between, and if there are any, homosexual voles and regular or heteronormative voles? Excuse me, programming, being any different? And also, will voles adopt another vole the same way a frog would?
Yes, you can swap the young of voles, and they will not seem to care. But whether that means they can't tell them apart, I wouldn't conclude that necessarily. But we have not looked at gay voles. Work around same-sex behavior in animals. The hypothesis I favor is that bonds generally serve these social functions. And one of the ways you can have bonds is through sexual interaction that engages these mechanisms. And you see a lot of same-sex behavior in other species in contexts in which bonds are really valuable. So that is my take on it. But I haven't seen any evidence that we have gay voles. Yeah. I haven't looked that hard, but.
So I think we human not only bond with others, but we also bond with other animals like cats. But I realize I talk with ChatGPT a lot personally. So I wonder, can we extend our attachment mechanism to intelligent machines? And might there be any evolutionary constraint or neuroscience constraint that limit our attachment to them?
Oh gosh!
Wow. Good luck. Good luck with that one.
That's a great question. I think we know the answer, right? And one of the answers is yes. I mean, people get attached to their chat bots routinely, right? People feel they have romantic relationships with chat bots, and so yeah, that's certainly possible. I think it's interesting to think about our relationships with other species. Like Lucy Brown, who I mentioned briefly, I had lunch with her in New York a couple of years ago, and she told me this beautiful story. She says, like, because that paper that I talked about early in the talk, showing that human brains and love got so much attention, it was pretty common for journalists to come and ask to be scanned while they were looking at photos and things. And so she had accumulated a lot more data showing these particular areas that were active. And she was older, and her husband had died, and she had fallen in love again. But around the same time, she had also gotten a dog, and she was crazy about the dog. And so she decided to scan herself with the dog and with her new boyfriend. And she was delighted to report that while they both had activity, they weren't exactly the same.
Hi, great lecture, thank you. I was just wondering more about the first time that they saw their mate, like how clearly were they able to remember this memory as like time went on?
Yeah, I mean, that is such a great question, and I think that's one of the things that we're most excited about studying. Like, now we have these whole brain maps. We have a grant that is being reviewed to try and make a variety of vole that would allow us to tag the neurons that are active and then later replay them or reactivate them remotely, minimally invasively. And we're hoping to sort of see like all the places in the brain memory might be, but we don't have a clue. Yeah, I mean, we have a clue from literature. We don't have any data. Yeah, come back, please.
Hi. Thank you for the lecture. It was great. I think a lot of human behavior is often thought of as metaphysical. For example, willpower. But research has shown that willpower is related to one part of your brain antagonizing another pathway in your brain. So, in your experience, has researching love as something mechanistic rather than metaphysical changed how you experience it?
All right. By the way, for my human biology students, she's my favorite. Not this year's class from a couple of years ago. That is such a fascinating question. I think I definitely think that thinking about the nitty-gritty of how love might work and why it works changes how I view my relationships and the relationships I see around me. That is certainly true. I don't think it cheapens it. I think it's like, appreciating a painting more deeply because you studied the pigments or you studied, the technique, right? Like understanding how it works makes it, like something that you can maybe more fully grasp. But for me, it doesn't lessen it at all.
Hello. I'm curious because you said you saw both sides of bonded pairs, and I wanted to know two questions. One, did you find that one gender experienced love more strongly than the other? And 2, did you ever see cases of unrequited love between the voles?
Those are great. Well, okay, so as for the differences between sexes, yes. And I think that is something that's really fascinating. I mean, it makes sense that sexes might differ in the value of a partner, right? And the nature of that difference might lead to some surprising things. Now, I haven't thought recently about sex differences in pair bonding and what it might mean functionally, but I do know that they exist. Like, males and females will work differing amounts to gain access to their mate. And if you separate a pair, males will wait longer before they're ready to fall in love again. Yeah. So, yeah, what does that mean? I'm not sure. It's something I've been meaning to think deeply about, and I haven't.
We have time for a couple more questions.
On the slide that showed the sections of the brain and the activity in different parts of the brain, the legend for that slide had 2 parts to it, and it was like romantic bonding, and it was also sibling bonding. Are the hormones related in sibling bonding? Like, how does it differ? And
That's so very insightful. I just gave the most superficial gloss on this experiment. What we actually did was to take animals that were sexually experienced and then pair them up, or handle them, screen them, do everything else we did with those animals, but then return them to their siblings, who they grew up with and they're close to. And then we looked at different time points after reintroducing them to what was going on in the brain. The time points were like a couple hours after they first started to mate. What happened at 6 hours? So 6 hours is like this point where the bond starts to really form, but it's not stable yet. And then by 24 hours, the bond will be essentially permanent so long as they stay together. And so what we did is we compared the brain activity in those pairs that were bonding to the activity of male or female animals that had just been returned to their siblings. And the reason there's no blue on there is that there was very little activity that was evoked by coming back to their siblings. Most of the activity that was unique was in the bonding animals, but the siblings showed a lot of behavior that sure looked like bonds. Like they would greet each other, they would make these little contact calls, which no one had noticed when they got farther apart. And so yeah, I do think the siblings have bonds as well. Yeah.
Thank you for the lecture. It was a really good lecture. Earlier you brought up how the voles prefer to be with the bonded versus the stranger. I was also wondering if you observed anything similar to if they showed distress or any decline in health if voles that were bonded were separated for too long,
Yeah.
if they were separated entirely.
Yeah, so that's a great question, and one of the many prairie voles basically have become this kind of major workhorse within animal neuroscience for understanding the roles that bonds play in all kinds of aspects of our experience, but in particular in health. And so, separating animals that have bonded has become a model of grief. You see a lot of activity that's similar to activity you might see in someone grieving the loss of a partner, a human. And certainly you do see that. And eventually the bond will decay. But they are generally, like people, they are generally stressed by being alone. That's why we house them with their familiar siblings. Unless they're mated, then we house them with their partners. But we never house them alone unless we're trying to understand the consequences of being alone. Yeah.
Okay, last 2 questions real quick.
So if we know that faithfulness correlates to kind of having better memory,
in the voles.
Yeah, in the goals. What could unfaithfulness have over time? What effects could that have on human evolution, possibly?
Uh-huh. Yeah, so I think, I don't think that the aspects of fidelity or infidelity or partnerships in human relationships are exactly the same as they are in voles. So I think the reward stuff is the same, I think. But I think the specific aspects of memory is less clear. Presumably the retrospinal cortex is involved. But the bigger question is about the consequences of faithfulness and unfaithfulness. And I hesitate to use those terms, certainly in the context of voles, because they're very culturally loaded. And honestly, my perspective on all of that stuff is very libertine. So I think if you look at all of human history and the geographic variation, we're kind of monogamous, all right? I mean, we form pair bonds, certainly, and rear young together, but those relationships take a lot of different forms, and they're not necessarily exclusive in a lot of societies. So I would be reluctant to make any... I guess I would treat with skepticism any claim about a relationship between fidelity and a strong moral judgment, which, anyway, we can talk more about that. But
all right, final question.
How does it live with the microscope on its head?
Yeah, I know. The microscope is very light, but it also is supported by a little, it's not on all the time. It's only on when they're doing the experiment, when they're actually recording for 20 minutes or something. But during that time, the cord that runs to it is on a little kind of tether to sort of hold it up so it doesn't exert pressure on the vole. They seem to do okay with them, but I wouldn't want to have one on my head.
Thank you. Well, I have to say, when no one had a question right at the end of this, it's that was a long pause, longer than we've ever had. I thought maybe after 140 Hot Science School talks, there'll be one with no questions. But instead it was the opposite. You all had some really amazing questions. Kudos to you. And kudos, thank you, Dr. Phelps.